Hello zoology students, this is Mrs. Draves. This is going to be our flip lecture for the scientific method. And I'm sure most of you know the general steps of the scientific method. I'm going to go ahead and write those down. So you should title this Scientific Method Flip Lecture, okay? So I use my own little kind of acronym for the scientific method. DMTAD. It's my own little acronym. The D stands for determine the problem. Okay, so with science, many times people just generally have questions. So after that activity that we did with live animals, it was called What is an Animal? You guys observed preserved specimen of animals, and you also observed live specimen of animals. And maybe some of the behaviors that you observed raised questions. And so then you start to try to answer those questions. So you would try to make a hypothesis. So you start to want to figure out why they do what they do. So, for example, if you looked at the crayfish and they were in a gravel substrate, so they were in a tank and there's gravel on the bottom, and then maybe you looked at crayfish that didn't have gravel on the bottom and you noticed that their behaviors were different, you might make a hypothesis that does gravel impact crayfish behavior? And so, or you could even be more general about substrate type and affecting crayfish behavior. So then you make a hypothesis that yes, the benthic bottom or the substrate that they are on definitely changes their behavior. And let's say you make a hypothesis that with gravel, their behavior is much more active. Okay. So then in order to test that hypothesis, you have to design an experiment to test it. So the T here actually stands for test the hypothesis. Okay, this is probably the most difficult or the most specific step-by-step part of the scientific method, because you have to design an experiment and you have to design it with lots of things in mind. You may have heard over the years in your science classes things in experiments called constants. Okay, and constants are all those things that remain the same. So you want to make sure in this particular example with the crayfish and the benthic bottom or substrate type that you have a couple of tanks set up, and so you're repeating that same setup. Maybe the amount of gravel that you have in there, the temperature of the water must all be the same, the lighting in the room must all be the same, the number of crayfish that you have in the tank must be the same. So those are just some examples of constants. And then there are things called variables. There's the independent and dependent variable. The dependent variable is the thing that changes, or what you're trying to see if it would change, and then the independent variable is the thing that you're adding to the experiment that causes the change. So in this example, the independent variable is the gravel type, or just having a benthic environment other than just the bottom of a tank. So you're adding the gravel to see what would happen. And then the independent variable is what would happen. And so that would be the activity level of the crayfish. So dependent variable would be how the crayfish behave in response to the independent variable, which is the gravel type. So then you also have to make a thing called a data table, within which you will record the data that you're collecting. And there's qualitative data and quantitative data. Qualitative data is more like what you see There are observations, okay? So you're literally writing down moving from side to side, or moving their callipeds, which are those large claws, or moving them up and down. Okay, that's like a description of what's occurring. It's kind of like you're using your senses to observe. And then there's quantitative data, and the word quantity is in there. So quantitative data, yeah, that's right. Quantitative data is numbers, like the number of times maybe they move, or measurements, okay, things like that involve numbers, okay? And then once you have all that data, you have to analyze the data. Okay, and so by analyzing the data, you're looking at what you stated as your hypothesis, and is the data supporting what you stated, or is it not? In a lot of cases in science, it's not. So you have to repeat the experiment over and over again, okay, to actually have quality data. And then you draw conclusions about that data, okay? When you draw conclusions, you write a paragraph, kind of like going over the whole entire investigation. The paragraph should be, 6 to 8 sentences long, just to give you an idea. I mean, not, there's scientific experiments where their conclusions are pages long. But in this case with zoology, I like to give students a framework. We're looking at 6 to 8 sentences. So what would those sentences be? Well, you would definitely restate your hypothesis. Okay, I'll put hypoth for short. You want to mention if you were if you were correct or not. You want to mention all of your data, and you actually want to mention data, like you want to specify data from your data table. So in that sentence, you would say something along the lines of referencing trial 2 during minute 5, that kind of thing, so that you can use that in a way, in a statement that supports if your hypothesis was correct or not. Then you want to identify error in your experiment. Okay, so error, what is that? All the things that basically would have affected the outcome of the experiment. So let's say during your scientific experiment, you observed trial 1, and then your partner in your lab group observed trial 2, and then another person in your lab group observed trial 3. That's an error, because you want to have the same, you want to have as many constants as possible. Okay, so maybe it's all of you observing. Okay, that would be fine. But when you're having different people observe each time, that could be a source of error, because not everybody has the same observation skills. And really, an error all the time is human error. We're not perfect. So maybe you looked down on the floor or looked at the clock really quick, and during that split second, there could have been movement in the crayfish and you missed it. The noise in the room could be an error. Maybe you didn't measure the water properly, so there wasn't the same amount of water, it wasn't the same temperature, there wasn't the exact same number of pieces of gravel in this particular example. So all of those things that could possibly have affected the outcome of the experiment that were not, constants, okay? And then you want to propose how you would conduct the experiment the next time. So if you were to do this again, what would you do different to make it more scientifically accurate? Okay, and don't just restate all the error in these last few sentences, because I want you to get in more depth about it. Okay, so don't just say that we aren't going to have we are going to count every piece of gravel. Just be a little bit more specific as to if you were to give advice to somebody investigating the same investigation, what would it be? Okay, so this is my little splurb of the scientific method, but let's look at this other presentation I have that will help us even better understand the process of science through this means. Okay, so the scientific method involves, stating a problem or a question, and that can be solved. That actually can be solved through experimentation. So when you guys do your investigation, you want to be able to answer a question through running an experiment. You make observations about what you question, and you formulate a hypothesis about it. You want to predict what the possible answer will be to the problem. So another example other than the crayfish is that if soil temperatures rise, then plant growth will increase. Okay, so when you guys state your hypothesis, you need to do an if-then statement. Okay, when you formulate your hypothesis. So if benthic bottom changes, then crayfish activity will increase. Okay, something like that. And then you experiment. And to do the experiment, you have to have a procedure. And this is really important because you want to be able to write your procedure in a way that somebody else can repeat it. So you want to be very specific. With that said, you also need to have a materials list. And so you guys will be getting a handout for your research project, and I kind of have outlined this a little bit for you to guide your thinking when conducting this experiment. You want to make sure that your outcome is measurable, meaning quantifiable. So that would be a quantitative data collection, but in our case you can also have a qualitative data collection. Okay, so just because this is just like an introductory experiment, and I want you guys to kind of have your own freedom with regards to what you're investigating. I mentioned earlier the independent variable. Again, this is the one that's manipulated. It's a factor that we vary in the experiment by the experimenter. So in the crayfish example, it was gravel. Okay. Another example would be John is going to use 25 grams, 50 grams, 100, 250, 500 grams of sugar in his experiment. So he's going to use a certain amount of sugar in the experiment to see, I think this was about bread, okay, and if it would increase the size of bread as you bake it. The dependent variable then is one that responds to the independent. So the independent is what you manipulate. The dependent is what you see change in as a fact of being exposed to the independent variable. So in this case, with the bread, in this example, it would be the size of the bread. One thing I forgot to mention in the last thing, definitely write this down, is the control group. So you want to have a standard for comparison. So this is a group that serves as a standard. So this one would get no treatment. So in the case with the crayfish example, it would be a crayfish in a tank with no benthic bottom. Okay, so there would be no gravel. In the case of the bread, then obviously that would be the one with no sugar. Okay, so the control group is your standard for comparison. The control group is exposed to the same conditions as the experimental group, except for the one variable that you're testing. And that's one thing that you want to make sure is that you are not testing more than one variable. You have to test one variable at a time. Okay? Your constants I mentioned before are all the factors that are kept the same. Okay? So with the crayfish example, we want to make sure that temperature is the same, the amount of gravel is the same, the number of crayfish is the same, all of that. With the bread example, it could be, the amount of ingredients that made the bread. Okay, so have the same amount of salt, same amount of flour, same amount of water, but different amounts of sugar. So the constants will be everything but the sugar. The trials, you want to do more than one trial. You want to replicate it, and you want to make sure that all the conditions are the same in those trials. Okay. And usually, like for this thing that we do in zoology, I want you guys to do at least 3 trials. Okay, so that's a minimum of 3. And then once you have your trials done, you collect and analyze those results. So you should have some kind of data table, and that's information for you to draw from that you collected. So this is an example of a data table. So you have your amount of sugar on the left and size of bread loaf on the right, and then you have the number of trials and the measurement. So this is quantitative data here. So you want to create some kind of data table, and please use rulers when you do this because it needs to be neat. And then, like I said before, with the conclusion, you want to state if you accept or reject your hypothesis, and then what I said before with my own notes with regards to the conclusion, doing all of these things here. So you want to restate your hypothesis if you are correct or not. You want to actually analyze your data, identify error, and then propose an explanation for the next time somebody would investigate the same thing. And that's what this is here at the bottom. Making recommendations for further study and improvements to the procedure. And then this, we probably won't have time to communicate our results, but we will, maybe we will, we'll see. Depends on how quick we get through this process, but this is a really important part of the scientific process, is being able to communicate what you found, and then having entertaining questions that are posed from the audience regarding your investigation. And so that's that. So this marks the end of our first lecture for zoology, and this will prepare you for your own group's scientific investigation. All of this information that you just listened to, I should have said this in the beginning, should be on the right side of your composition book. And then I expect you to analyze those notes on the left side with the way that you interpret the whole process. Okay? So remember, the right side's input, the left side's output.